There we go.
Thanks for the opportunity to present my work here. My name is Chunlei Liu. I am a research officer from the Computational System Biology Group at the Children's Medical Research Institute. Today, I will share our work on multitask learning from single-cell multimodal omics with Matilda. First, give a brief and quick introduction. Matilda is a multitask learning method for integrative analysis of single-cell multimodal omic data. A key observation in Matilda is that many common tasks in single-cell multimodal omic data analysis are closely related to each other. Based on this observation, Matilda exploits the data simulation, cell type classification, dimension reduction, and feature selection in a single unified framework by exploiting the interrelationship among tasks. This is our framework. We use a variational autoencoder here. The input is a multimodality data, where the green and pink color represent different modalities, and we encode each modality separately. Then we have a joint learning layer to study the information across different modalities. After that, we decode each modality separately and obtain the reconstructed data for each modality. This is our task 1, single-cell multimodal data simulation. And we introduce a classification branch after the latent space. This is our task 2, cell type classification. These two tasks are trained simultaneously. The results of task 1 can be used to do a data augmentation and balancing, which helps to get a better result for each task. After the model is trained well, we can use the latent space to perform a multimodal dimension reduction and visualization, and use a gradient descent-based feature selection method to select the markers for each cell type. Matilda now available at GitHub in Python. Welcome to try and use. Then begin to introduce how to use Matilda. First, introduce the installation. We use Conda environment. We provide 2 ways to build the Conda environment. One way is you can build it by yourself according to the GPU version. And another way is we provide an environment file. You can directly install this file to build the Conda environment. And then come to the usage of Matilda. We first train the network using the following command. Python main Matilda train.py, then give the data path as the parameters. Because we use matched multimodality data, so we give 3 data path parameters, AEDT, and attack. If you only use the statistic data, which contains I and ADT only, then you can just give the path of I and ADT, leave attack as default, none. And then after the model is trained well, we can perform different tasks. We use this file, main Matilda task.py, to perform different tasks. For the simulation task, we can set the simulation as true, and then set the cell type index and the number which we want to simulate. And this is the visualization of the results. The first row is the real data. The second row is the simulated data. For the right one is the anchor cell type. We can find that the simulated data is very similar to the real data. And the second is the cell type classification. We use the same file to run, but set the classification parameter as true. Then we can predict the cell types in the query data. The output format is like this: the cell ID, real cell type, predicted cell type, and the prediction probability. And the third is dimension reduction and visualization. Similarly, we use the same file, but set the dim reduce parameter as true. Then the dimension reduced data can be saved in the output folder. After do you map on the dimension reduction data, we can obtain the results like this. And the last part is the feature selection. We just need to set the FS parameter as true. Then the gene importance score for each gene and each cell type will be saved in the output folder. And based on this results, we can do different visualization. And I would like to thank for all the members in the computational system biology lab and the Sydney Precision Bioinformatics lab. That's all for today's presentation. Thanks for your listening.
Thanks a lot, Chunlei. Is there any question in the audience for the speaker? Yes, we have one coming from Giovanna.
Hi, Chunlei. Sorry if I completely missed this in your talk. You said that the data simulation is part of your multi-omics integration framework. Can you elaborate on that?
You mean the data simulation part?
Yes, yeah.
The data simulation part is from the input is a multi-omic data, and we use a VAE, variational autoencoder, to reconstruct the data, because we use the variational autoencoder. So in the process, we have some variation. So the output data will be somehow different with the input data, but has the same characteristic. So this part we regard it as a data simulation, but actually it's more accurately it should be the data reconstruction.
Okay, thank you.
Is there any other question for Chunlei? No. If there are no other questions, thanks again a lot for your talk. Please, yeah, Jeremy.